My name is Ian Stocks. I'm a taxonomic entomologist with the Florida Department of Agriculture and Consumer Services, Division of Plant Industry in Gainesville, Florida. The groups that I'm responsible for are the scales, mealybugs and their relatives, and the Allarodidae, which are the whiteflies. Okay, well let's go ahead and we'll finish up then with Philephedra tuberculosa, because that'll take us to another part of the key. Okay. So again, we'll drop it back down to the 4x. Okay. All right. So let's assume then that the slide we had was not Cystitia, and we've gone to couplet 3, the second part, and we've decided it's not as above. Okay. Which means we've got to drop it to 4. Okay. Let me just make sure. Here we are. Okay. But first, let's just give a kind of a general tour of this one to get our bearings on where the characters are going to be found. So we're going to start at the top. Okay. The first thing is the marginal setae are really quite pronounced in this species here, this specimen. Not just the species, but this genus. They're all quite stout and pointed. And they are uniform too. If you follow the margin around, you pick a side that's got a nice spiracle. Okay, spiracle there, spiracular furrow. They're very similar right there in the marginal furrow. Okay, they're not a very divergent morphology from the remainder of the marginal setae. So again, there's the. And let's see which plate does this one correspond to. This is plate 6, and the marginal setae and spiracular setae are in figures 4 and 5. Okay. And here's the posterior spiracle in this one. Spiracular furrow. There's a spiracular margin right there. Okay. All right. Okay. Antennae are fairly straightforward in this genus, as are the legs. As far as I know, all the species have a tibio-tarsal sclerosis, and they're well developed. Go down the body again towards the anal plates. Go up in magnification on the anal plates. It's really not a whole lot interesting in terms of key steps going off here, because you will have wound up in the part of the key where they're just going to be classified as quadrate, as opposed to some other more aberrant morphology. So this is just another example of the quadrate morphology here. Okay. Now, pick a part of the slide where you can see the margin quite clearly with the marginal setae, and then just go on into the interior a little bit. At 10x, you're really not going to be able to perceive much. But in that general region, if you go up to 20x, you can see that, again, there's regions where the cuticle is changing kind of color a little bit. These pale spots in these fields of in this case, pink. And each one of those is associated with another structure. Okay, so this is a fairly low magnification, another type of tubular duct. All right, so find a good patch of those, and then go up, if you can, to the 40x. Okay, and get a bit more detail on the morphology there. So you see they're quite complex. Again, you should be able to perceive, at least in if they've got the right orientation, where the duct opening enters onto the cuticle. You'll see the whitish patch. an inner tube, a sclerotized cup-like structure, and then another much less prominent kind of tubular projection ending in a fairly indistinct gland. And the comparison structure there is in figure 3 of plate 6. Okay. For the genera that I see, the morphology of those ducks right in this region right here, what they call kind of a cup-shaped invagination, is characteristic for this genus. This is Philephedra. Okay, so if we go to the key, all right, let's see. Okay, what we're going to do is go back out on the magnification a little bit so we can pick up the steps in the key here. Okay, so we're on, we've got step 4, ventral submargin with numerous tubular ducts often forming a band. That's the first statement in couplet 4, and that's exactly what we just saw in that last series of images, was that band, because you could go almost anywhere on that submargin on either side and be in that band of ducts. So we're positive for that character. Now we can go to 5, key step 5. The contrast character for 4 is an absence of submarginal ducts. The genus Coccus, which has the brown soft scale and the green soft scale, the species Hesperidum and Viridis, very, very well-traveled scales, very common, very polyphagous, in some cases very pestiferous. That's actually a genus that's fairly depauperate with tubular ducts. When they're present, they're almost entirely restricted to a region just on the interior of the legs at the bases. Okay, and this makes sense because if you look at these animals in the field, they're almost naked. They produce very little wax. The extent of the wax they make is just really just a very thin film that covers, and it's glassy. It just barely covers the body, so you get a full, you can see the body quite clearly. So, and that, of course, then is going to correspond to a lack of structures that produce that wax. Okay. Okay. So now on key step 5, submarginal tubercles absent. The key where we are now for step 5 is going to ask us to look for a character called submarginal tubercles. Okay, whether they're present or absent. Now, the submarginal tubercle we saw earlier in the general tour of morphology, and it's basically just a circular mound. It's clearly got three-dimensional structure to it, but it's just basically circular in outline, and it's restricted to the margin. Okay, there's never very many of them, even when they are present, but there's usually going to be, 1 or 2 or 3 or so along the margin. Now, if you look at the slide that we have here, and it's fairly straightforward because all you have to do is start near the head and look at the marginal setae, which are very distinct, and you can just scroll through the entire slide, and you're not going to find the dorsal tubercle. Now, the thing you will find, okay, and I'll get one in focus here. Even at 40x, it's going to be fairly prominent in its square footage, so to speak, because I've got right here in the margin. I'm just not getting the right kind of contrast to make it. Yeah, I'm going to have to go up in power here. I'm not getting the right contrast for it. There we go. There we go. Okay. So we're right here. They're going to be fairly prominent at this kind of magnification, really just disc-looking structures. Okay. All right. Now they are on the submargin, but you'll see that they are elsewhere on the dorsum also. So there's one there. It's also on the submargin. There's one there. Now, I want you to compare them to the illustrations on plate 6, which are figures 6, 7, 8, and 9. Okay? See if you can find one that looks like it's in a nice clear area and you're going to be able to maybe get a good, more detailed view of. So, okay? So I've got this at 10x, okay? Now I can go up to 20x. All right? And let me see if I can improve the... All right. Again, I focus in and out a little bit there. You can see it's in figure 6, for instance, you can see at the very bottom of that illustration, there's that the round area. Then there's a sort of a conical area, and then there's almost like a little flagellate area, the little tip of the interior of that duct. Okay. So you can pick up on that. I can just make it out on my slide. If I've got this one right here, the duct, the very fine tip of the duct starts about right there, connects to the tip of this, and then goes to this structure right here. Now it becomes even more clear what's going on if you're able to crank it up to 400x. You get a nice face kind of going off here. Again, there's the flagellate tip right there, the interior. And if I try to find another one, the opening of that sort of mouth type opening of the tubercle has in and of itself a fairly complicated morphology. It's not always easy to see, but it is a species level character in some cases, what the interior of that larger opening looks like. And I'm having a hard time finding one that really works here. But you can just about perceive in plate 6, figure 9 at the bottom that it does have a fairly complex morphology there. It's a fairly strongly three-dimensional structure, so it's hard to get a good picture of all of the components at one time. Now, for the instances of this key, you've determined that that morphology is consistent with the inverted duct tubercle, so you've got Philephedra. One of the characters that's relevant at the species level then is determining the distribution of determining the distribution of those. Okay. This will be our last character discussion here. If in your field of view, if you kind of start either at the front, the anterior, the posterior, and then keep yourself restricted to say the left side or the right side of the animal, and then do a count of those, okay? So you add them up, go, okay, 1, 2, 3, 4, and keep track of where the distribution is. The different species in the genus Philephedra can be keyed out by comparing if those duct tubercles are restricted to certain areas of the dorsum, or if they're not restricted, what's the number of them in their distribution? Are there just a few of them? Are there many tens of them, for instance? Okay. All right. So, all right. Okay, so that brings us to the end of the key.